Hello, everyone. I would like to welcome you to the fourth webinar that's hosted by Progenesis Academy. As many of you may know, Progenesis has launched COVID IVF, which is the first PCR test allocated for fertility patients and staff. Last week, we have also launched the first ELISA antibody testing. So since then, we've received an overwhelming number of questions in regards to the different types of tests that are currently available and the differences between the testing. So we wanted to provide a comprehensive overview of real-time PCR, antibody testing, and rapid tests. We're hopeful that this could provide guidance for fertility centers as they open back up or as they continue to see fertility patients during this outbreak. We're going to start off with a 35 to 40-minute presentation followed by a Q&A session at the end. Feel free to use the chat button or the Q&A for any questions that you may have, and we will address those to our speaker. I would like to introduce our speaker, Dr. Nabil Arrach. Dr. Arrach is the scientific director here at Progenesis, and he had a big role in launching both our PCR and antibody testing. Without further ado, Dr. Arrach, the floor is yours.
Thank you so much. Well, today we're going to be talking about diagnostics and options for laboratory to use either RT-PCR, antibody, or rapid tests. So this presentation will let's start first by talking about COVID-19. Last few weeks, we have been talking about the coronavirus, and I mentioned in my presentation last time the genetic makeup of the virus. Just a quick reminder, COVID-19 belongs to beta coronavirus that has a high similarity with SARS virus and some similarity with the Middle East coronavirus type. We have gone last time through the presentation on the how the virus infects. We talked about the S protein role in adhesion and penetration, and how the virus replicates itself. And as a little bit of a kind of summary of what we have said before, so the virus has an open reading frame, 1A and 1B, and that is the first open reading frame that gets translated first, and that play a role as a replicate enzyme and does play a role in the a replication of the RNA and the translation of that RNA into peptides. And so the three main components of the virus is the, in yellow is the S protein that is used for adhesion. And then you have the 3' polypeptide that is used for packaging and release of the virus outside of the cell. And then you have the 5' that is used for replication. So when you look at the genome of coronavirus COVID-19, it has approximately 30,000 base pair. For a virus like this is really a big genome compared to other viruses. Most viruses are below 20 kb, 20,000 base pair. It has 14 open reading frame and 27 proteins. And so today we're going to be talking about the tools that are available for COVID-19 testing. And for example, the first option we're going to be talking about is how do you screen for the virus? And so the virus is normally screened using symptoms, right? You have to use respiratory symptoms, cough, fever, temperature, And I will show you guys a study on the frequency of those symptoms in affected population. So just to give you an idea, that is a quite relevant screening method. And so the CT scan is used as a way of diagnosis. Then you have the RT-PCR, and you have the antibody testing. And within the antibody testing, we have the rapid test, and we have the ELISA test. And I will explain the differences going forward. So going to the screening of symptoms, if you look at this study, so we have a large study where they're looking at what symptoms can be identified in patients. And this, if you look at this table, 88% or more than 88% presented fever, high fever. And what that means is that if you have a patient that you suspect has coronavirus, their fever is more likely to be high, right? 67% have cough. So you can still use symptoms. That's a good way of screening, but eventually you're gonna have to rely on genetics to be able to have an accurate prediction and diagnosis. So the genome of coronavirus has a number of proteins. The one that is currently or heavily used for diagnosis is protein N. That's and when we test for RT-PCR, we use RNA, right? We use primers and RNA. And those primers that are designed for N proteins are the ones that were originally approved and recommended by CDC. Down the road, we have seen companies that have been bringing diagnostic tests with targeting more than one protein. For example, the test that we provide at Progenesis is targeting both open ready frame AB, N gene, and S gene. You can see three targets on the same on different genes, right? So you have to pay attention to what your test is covering as far as genome. And the reason behind that is that if you happen to have mutations, and we know I've spoken last time about the genetic diversity of the virus, and that's up to date, we have seen more than 229 mutations in coronavirus genome. And so you don't want to rely on one single probe and one single pair of primers, because you may be missing certain isolate that have mutation on that site. So that's why I like the approach of having multiple gene sampled at the same time. Also, to avoid false negatives, if you have multiple targets, you will be able to at least identify one of them. Say if the target against S protein does not work, the N would work, or the one against open ready frame AB will work. And that minimizes the chance of having a false negative result. So as I mentioned earlier, the number of mutation in coronavirus genome is pretty high and is increasing. It is increasing with time. I put here a sequence just to give you an illustration of how this mutation can affect the performance of the PCR. Let's say you have a genome here where a mutation occurs in these RNA sequence that will be retrotranscribed into cDNA in this case. If the primers have that mutation on that, if the primer lands on the site of the mutation, so that maybe that primer will not be able to amplify throughout the genome. And what that means is you have a failure of amplification. Even if you have internal control for other phage, they may perform normally, and you may have a false negative result. So how does real-time PCR work? So first of all, you have to extract the RNA, the viral RNA. Extraction RNA is fairly easy because coronavirus envelope is very sensitive. You can lyse it pretty easily, so there is nothing special there. So when you have an RNA, you have to do a retrotranscription to cDNA. So you have to obtain a cDNA, which is basically converting RNA to a complementary DNA. And then the PCR will kick in to multiply that DNA into a very large number. So the two components that I use in the PCR are the primers. The primers will amplify the genome, and the probe will give you the fluorescence. So if you look at the right-hand side, you see this probe right here. So the probe would land into the amplified DNA. If the probe is stretched, then the fluorescence will come out. That means you have DNA there where the probe is attached to, and that's what the fluorescence will come out. If there is no DNA there, the fluorophore of the probe and the quencher will be too close to each other. The quencher will stop that fluorophore from fluorescing, and then you don't have a fluorescence. As you amplify your DNA more and more, there are more probes that are stretched, and therefore more fluorescence over time. So if you look at this figure here at the lower end corner, you will see that, for example, this just represents on the x-axis the number of cycles. So you have 0, 2, 4, 6 cycles all the way to 40 cycles. Those are cycles of amplification. And on the y-axis, you just see the increase in concentration of the DNA. And what you see, for example, in the red, you can see that is an exponential, because the DNA multiply exponentially. But when you take the value, the median value, you get a 15 cycle on the y-axis. All that means is after 15 cycles, you start seeing a exponential increase in DNA. So we use the CT cycle to define how much DNA you have amplified. For example, if you go to the blue or the purple curve, you can see that the median here is 37 cycle. That means the DNA, that sample needed a lot of cycles to be detected, and therefore there is not too much DNA in the RNA in the sample. So that's how we quantify RNA in a sample, by looking at the CT cycles. Less CT cycles mean you have a lot of RNA in the sample and therefore a lot of viral load. If you have very low CT, very high CT, that means you have very little RNA and therefore very a small viral load in the sample. Now, if you look at the studies, and we get a lot of questions, what is the accuracy of the RT-PCR test? Or what is the sensitivity? And what is the specificity? And my answer has been always very similar. You have to look at the context. You cannot just say, well, this is specificity and this is sensitivity. You have to look at how that study was done and analyze it before you go into the conclusions. So for example, I'm giving you here an example where the real-time PCR have shown here, they said it was 61 to 68% sensitivity, and that's with nasopharyngeal aspirates. And when you look at the method that they have used, it wasn't a true RT-PCR. It was an RNA that was with retrotranscription to cDNA, and then instead of putting a real-time amplification, all they've done is a 2' amplification, and they run a gel. So that's not an RT-PCR reaction, right? So really, you have to look at the context before taking a conclusion of what specificity and sensitivity you're dealing with. So as far as sampling methods, they are the gold standard for sampling COVID-19 is nasopharyngeal. That's the method that has been validated the most, have used extensively across many studies. And then we've seen other type of samples that have come out in the markets. One of them is oropharyngeal. Now we have seen sputum or saliva. And this study is basically comparing the nasopharyngeal data or sensitivity to saliva sensitivity. And so they have taken patients and collected this, two different samples from the same patient, saliva and collected the nasopharyngeal sample, and they collected it every 3 days, and they waited through the course of infection, and then they compared the results. And basically what they are saying in this paper is that the sensitivity of saliva seems same or even better than nasopharyngeal. The issue here is we know nasopharyngeal sample is good. We know it's high quality. We know it's targeted towards the type of sample we wanted to detect. The problem with nasopharyngeal sometimes, you really have to know how to collect the samples. Because if you do not perform the best technique for that, then that's my dog here making noises. I apologize for that. So if the nasopharyngeal sample is not performed well, then you may have a false negative. And so it's important that you perform the sample the best way possible. And the saliva sample, the beauty about saliva sample is its consistency. You know, it's very hard to get wrong samples. So when you try to collect sample from a patient, then most of the time, all the time, they're gonna give you a good sample. So it's more consistent across different testing phases. And so there are other studies that have looked at the correlation between RT-PCR and chest CT. And in this study, they wanted to show how good CT is as far as a diagnostic tool. So they used RT-PCR as a reference, and then all positive candidates from RT-PCR were tested with chest CT. 97% of them were positive with CT. So the correlation, when you look at the positives with RT-PCR, 97% are reproducible when you look at the chest CT. Now the negative group is another story. So they have seen that they have classified the negative into two categories: the one that are highly likely and the one that are probable cases. And with the highly likely cases, they have seen that 48% of them were CT positive, but they were negative on real-time PCR. So let's move to another type of test, which is testing for antibody. And antibody testing, obviously you have the IgG and you have the IgM. And this graphic, this is just an illustration, is not a real graph data, but it's very close to what we have seen, what we have collected so far as society. And so when you look at the blue bar or blue line, that's the RNA or the infection. And then you look at the IgM, So the infection lasts about 30 days. If someone gets infected, it takes them approximately 30 days to get rid of the infection, if they do, right? The IgM peaks at 2 weeks. In 2 weeks, you have a peak of IgM. So if you're trying to get IgM, so that's where you get it, 2 weeks. And IgG peaks at 4 weeks. So IgG is a more of a long-term antibody, and therefore, if you want to really get the good answer, you should go with IgG, and you should wait for approximately a month after the first exposure or symptoms. And so how does the test work? This is ELISA test. So ELISA test is a test that is performed in the laboratory. This is not a rapid test, right? It's a laboratory, it's not a rapid test. This is a laboratory grade test. The way it works is you work with plates that have multiple wells, just like the wells that you guys use in IVF testing, in IVF lab, but they have, those are 96 ELISA tests, ELISA plates. The wells of each, the bottom of the well of each, the bottom of each well is coated with an antigen. So there is an antigen that will recognize the antibody. The antigen is all across the plates at the bottom. So when you extract the blood from a patient, you have to spin the blood to take the serum. That serum, if it has antibodies, and you pour them, you pour that sample into the well, then the antibody will go and bind to the antigen. Remember, the antigen is at the bottom of the well, right? So you see this well right here, that's the antigen. So the antibody will bind to the antigen. Then you have to wash. You have to take off the supernatant, the serum, add wash solution and wash it a few times. And then you have to add the conjugated antibody. So that conjugated antibody is produced in a different animal. You can have sometimes mouse or rabbits or chicken. There are several species that are used for producing the conjugated antibody. The conjugated antibody is called conjugated because it has an enzyme attached to it. And so when you throw the conjugated antibody into the well, it will find its way, bind to the antibody itself, and stay there. And then you can wash off all non-binded conjugated antibody. Once you add the substrate will be used by the enzyme to be converted into a substrate that has color, and therefore lead to an absorbance at 450 nanometer. And that's what you see in yellow color. So it's almost like a colorimetric assay, but you're not relying on color, you're relying on absorbance at 450 nanometer. So you need a plate reader that will read that absorbance, right? And so that's how the test is done in the lab. The test takes approximately 4 hours. It's a long process. It needs always to use a duplicate, at least if not triplicate. You have to use a duplicate to compare the same sample twice. So, and you have to use a lot of validation involved in the test. You have to look at what is the, in terms of positive control and negative control, you have always to use two. You have to define that negative control, at what limits will be the negative control will be rejected, because you can't have a negative control that give you a high read, and you can't have a positive control that is giving you low read. So you have to define what those values are. So that's why I emphasize that if you really wanted to do antibody tests, you have to use a lab, because the lab their job is to run all these quality control, quality assurance, to be able to give you data that is most likely to be useful as far as diagnostic value. So this is an example where you look at validation that was done with the IgG test that we currently offer. So there has been several stages of validation. The first one is clinical trial. It was conducted by the manufacturer, where they have tested 30 patients that are positive with RT-PCR, retested with IgG, and 54 patients were negative with PCR or believed to be normal, and then they were tested with IgG. All these 30 positive were confirmed to be positive. The negatives were converted to be negative. On a second study that was done by the same manufacturer, you have the samples that were recovered from US patients on the right-hand side. And you can see here that 25 samples, I apologize, 25 samples were tested. Those samples came from patients that are affected, and they were all confirmed by RT-PCR. And when they were tested by IgG, 23 out of 25 were confirmed to be IgG positive. And that led us to a sensitivity of approximately 92%. At Progenesis, we have also tested serum samples coming from coronavirus positive patients. And that's what I represent here in the left, in the down left corner. And you can see the samples in yellow color are the ones that are known to come from positive patients. They came all positive. And the ones that are negative are volunteers that are healthy, that have not been exposed to corona or not aware of being exposed to coronavirus. And we tested them as well. And you can see that the values in blue, those are unaffected patients or volunteers, and they are below the control or around the control. In white is the controls. All these are duplicates, and we have a positive control, which is the one in green. So this is what we have done at Progenesis as far as testing. We continue to get more samples to validate and also to improve the sensitivity and specificity of the test. This is an ongoing process. The more samples we get, the better idea we have about sensitivity and specificity. This is a study unrelated to us, a study that was done to compare S with N targets. S is the S protein of the virus. That means that the antibody is designed for that protein. And the N protein antibody is designed for N protein of the virus. So what they have seen here is they have seen that the sensitivity of the antibody with N was much better. In this case, it was 94.7. They also compare IgG with IgM. So I mentioned earlier that IgG is a long-term antibody, but also when it comes to sensitivity and specificity, it's much better than IgM. It's a much better detection. And so in this study, you can see that when they have looked at, I think the number here is more than 2,000. It's 148 versus 95. So the bottom line is that the IgG value is always higher. So if you look at that line, that is 0.5, that is the OD of the sample. So above 0.5 OD, you see a lot of samples with the IgG coming higher than 0.5. That means the sensitivity is much higher. Most of your samples, if they are positive, you will see them. Versus IgM, they tend to give you less read, less intensity, and therefore just the sensitivity is reduced compared to IgG. So basically this study shows you two things. IgG is better than IgM, and N protein is better than S protein. And at Progenesis, we're using the full N protein as a target. So let's talk now about the rapid test. So the rapid test uses a very similar concept than the traditional ELISA test, with the exception that instead of running it in a laboratory with equipment and high quality material, you have a little cassette that is printed with antibody, conjugated antibody and antigen, so it allows you to detect in a matter of few minutes. I will show you guys the concept first, and then we can talk about the sensitivity and all that. So the cassette looks like this. The line that says C is control. So the C is printed with a, the printed line here shows an antibody that is printed on the line C, and that's antibody against a separate antigen, and we could talk about that in a second. Let's focus on the T, which is the test. T is printed with a conjugated antibody. So our antibody that we produce, we can make a conjugated antibody that will recognize them. And those conjugated antibodies are now printed, one for IgG, one for IgM. And they're printed just like a regular printer. So there is a machine that the antibody, the conjugated antibody, and you can run them through a membrane, and they will mix it will actually print the actual conjugated antibody on that membrane. And so in green, those are pads that will just absorb the liquid. So what happens, and on this lower side, you have the antigen that will recognize your antibody, right? So when you load on this little circle, when you load your serum, if you have any antibody, you are loading the antibody there. And the antibody will now bind to the antigen. The antigen is labeled with gold colloidal or colloidal gold, which is just a label that will emits color when the antigen, the antibody, and conjugated antibody, all three are together, bind together. And so when you load your sample here in this little hole, you're gonna load the antibody. The antibody will go and find the antigen right there at the very beginning, and it will drag it through the membrane. And when you drag it, it will pass this line. If it finds its match, it will bind now to the conjugated antibody. The conjugated antibody is waiting here in the line. So when this complex is formed, there is a color. And that's how you see if it's positive or it's negative. So what is the issue with the rapid test? So one limitation, clear limitation of the rapid test is the sensitivity. And we can go into the next slide where we can talk about the sensitivity. And so if you look at these study, they used colloidal gold assay to look at the sensitivity. So they have used 49 individual, 20 were tested positive by PCR, repeated PCR. And so what they found is that only 8 of them came positive. Only 8. That brings us to 36% sensitivity. So that means that many positive go undetected, or and that's will be reported as negative, but they are false negatives. And so in a separate note, there was a lot of testing going on in Spain, and they were finding they were using those little cassettes, and they found that they were not accurate enough. So I think they reported in newspaper in Madrid that they had some close to 30% sensitivity, and very similar to what we see in this study. So sensitivity is important in a test, in diagnostic test. If you are doing testing for your patients, would you give them a rapid test, knowing that it may fail, and 70% of the time you may fail reporting the right result? And so a rapid test is good for certain things. If you're going to do a triage that is not health, is not going to involve a clinical decision, a major clinical decision, maybe it's fine. But if that is a diagnostic tool that you are using to make a decision on, a clinical decision on, then you shouldn't be using a rapid test. Especially now with this IgG test, it can be returned within a day. So when we compare these two tests, the ELISA test and the rapid test, one is reporting at least 92% sensitivity. The other one is in 30 to 36, according to what we have gathered so far. The first one is performed by a laboratory professional staff and scientists. It has gone through a lot of validation, has gone through a QC, quality control, quality assurance, and then performed with the equipment, highly sensitive equipment. The second one is performed by a consumer. Even if that reached your clinic, you're gonna be using a product that was designed for consumers only. 50 minutes test is good, short. There is no really strong validation behind this, although there are a lot of, the way I see it is more of a manufacturing play rather than an actual laboratory test, because now there are some companies that are printing those chips, 100,000 chips a day. And so what is that quality that we're talking about? When we look at price, obviously the chips are maybe within a $2 to $5 a chip, so it's not very expensive. A test can cost $150 to $200, so it's substantially higher cost, but then you're gonna get a better accuracy and sensitivity. One of the most frequent questions we get is, what is your sensitivity and specificity? Whether that is for PCR or for IgG and IgM. And my answer to that was, you have to look at the context, and that's what I mentioned earlier. So when you offer a test the first time, whether here or somewhere else, you don't have that, you have an original or initial validation, clinical validation, a study, and that study is always based on a number of 100 patients, or close to 100 patients. You're lucky if you could get hundreds of patients in a study to release a product. And what happens with that is that you really don't know exactly your specificity and sensitivity till you test thousands. So what we report today may slightly change in the future. And so the sensitivity and specificity is not just related to the chemistry that you use or the test that you use. It's related to the accuracy of sampling method. Are you using any sampling method that is leading to high rates of errors? It's related to the stage of infection. Are you sampling the sample early on during the infection phase that is too low to be detected, or not even there, it's there, but it's a very low dose. And so, yeah, that's why the context is so important. You can present a validation in the laboratory, but when you translate that to real life patients, the things may change slightly. So when you have a patient that is coming to your facility asking for an IVF treatment, how do you screen them? Obviously, we have talked about screening with symptoms, and symptoms is obviously a very good thing to do. You look at the fever, the cough. As I showed in my presentation, I think more than 80% or 86% did have fever, patients that are positive. So it could be a good screening method to do triage, right? If someone has all the symptoms, we may want to put them on hold, give them 2 weeks or 3 weeks to do self-quarantine and then bring them back. If that patient is asymptomatic, what do you do? So in this case, you could do an RT-PCR. An RT-PCR, if it's positive, that means that the individual has the virus. It's asymptomatic, but has the virus. And so now become the decision of, do you want to take them through fertility treatment? You don't know the impacts of that infection down the road. We don't really understand the relationship of infection and pregnancy. That relationship, we don't know it yet with SARS. So that's a clinical decision you have to make. If that patient is negative, then you have two choices. You can go and look at their antibody and see if they have been infected in the past. That will give you an idea if they have been exposed. If they are positive in IgG, then that's a green light. You can go and proceed with the cycle. If they are negative with IgG, then mean they are negative with RT-PCR, no active viral infection, negative with IgG, no antibody, then mean that they are still prone to infection, and that's how you should monitor them. You know, you can monitor them as they come, every time in your practice, or every other week, or that's completely up to you and how you see the prevention work for your patients. And obviously, you can also I think the CT scan is done for those are already affected in hospitals. And so as I have made this clear in my previous presentation, we at Progenesis, we are trying to help. And we are trying to help in two different ways. One is to support patients, take the safest pathway to treatment by enabling them to get tested and monitor their status as far as infection. And also we wanted to help the clinics by reducing the impacts on the operation by allowing their staff to get tested. And we have seen clinics that wanted to test their staff using antibody testing. So the two tests that we currently help the clinics with are real-time PCR and antibody testing, IgG and IgM. We think IgG is the right way to get the staff tested and patients if the clinical decide to. With this, I am coming to the end of the presentation. I appreciate your time and patience. We're gonna release the poll now. We're gonna take some of those answers and share them with you guys. And after that, we will take some questions from the audience. And I will be here to address any concern, questions. Thank you so much. Miriam, maybe you can take it from here.
Yes, so while everybody's answering the poll, we can get started on the questions. The first one was from David asking, do the approximately 150 mutations we discussed several weeks ago affect the accuracy of the antibody test?
That's a good question. Excellent question. We don't know. The antibody assay that we have provided or we are providing has recombinants up to 6 different recombinant proteins. And the reason for that is to try to capture as much diversity as possible. Usually when you have a protein that you have an antibody against a long entire protein, you are more likely to find a match. You're more likely to have binding, even if there are mutations. If you are targeting the a peptide, a small peptide in the genome, then then you may have that issue. Let's say you are targeting a very tiny peptide and you have a mutation that will confer or will lead to a change in amino acid. Maybe that peptide will have a different configuration that will not be recognized by the antibody. But in our case, we're working with full N protein as a target.
Next question was from Carmen, asking if the rapid test tends to have more false positives or false negatives.
If the rapid test?
Mm-hmm.
The false positives does not seem to be a big problem in testing. It's more about false negative. So the sensitivity is related to false negative. That means a patient is affected and you cannot see it. And that false negative is what led to a low sensitivity in this case of the rapid test.
Next question asks, how many viral genes does the qPCR test analyze?
Our test is designed to capture 3 different sections of the viral genome. We're capturing protein N, protein S, and open ready frame AB. So we're targeting 3 different places for that. Some other tests are targeted towards N protein alone or N gene alone. So you have to look at each test configuration and ask what are the targets on that assay. It's important to know if you're targeting multiple places or just one place in the RNA genome.
Next question is from Fred Miller, asking if IgG can't be used as a method to confirm patient has immunity, since we don't know much about reinfection yet.
Yes, well, that's a big question. Do we know if it's IgG positive, that means it's immune? It may be immune. Experts in allergy and immunity says that they may have level of immunity. How much of the immunity, we don't really know, but we know that they have some level of immunity. And if a second wave is very different from the first one, and we have seen in our latest presentation on corona genome that there are different subspecies that are circulating, or type, I would call them more type. In the US, is slightly different from Europe, slightly different from China. And so if there is a second reinfection back in the US, for example, we may see a different type that we have not seen. But all this is it's literally just hypothesis. What we have seen in Korea, South Korea, that some cases of reinfection have happened. We don't have a lot of cases to base our data on, but it's a possibility.
And next question is from Ben, asking if there's any chance of improving the sensitivity of the rapid test.
Always, there is always a chance. But an inherited limitation of a rapid test, whether that is for colloidal gold or any other test, whether it's lateral flow or others, when you have a very rapid test, you have to assume that the sensitivity would not be the same as you are running a conventional test. Whether that sensitivity can improve, yeah, obviously it can. It can be a multi-detection system run on the same time too, where you can look at not just one target one antigen, but target multiple, or have an approach that is slightly different. But as for now, the sensitivity is not that high in the rapid tests that we currently see in the markets.
And with that, I believe that concludes the questions we have, and we've also closed the poll, and we can go over those results with everybody. So it looks like the majority of attendees have found that this webinar has helped them understand the different diagnostics options available for COVID-19, as 95% responded yes to that. 79% said that this was also useful in understanding the differences between the molecular tests. And then we've also asked how important COVID-19 testing was to your organization. And this was split with 61% thinking it was very important, and 34% thinking somewhat important, and 5% thinking it wasn't important. And then the last poll question was, what diagnostics options will you likely use? So the majority would have chosen ELISA test as the option that they will most likely use, 47% choosing for ELISA test, followed by symptoms, 45%, and then RNA was 41%, and then rapid test was 13%. And then most importantly, we also had an option for all of the above, and 33% of attendees have chosen that, seems to think that all the above are options they will most likely use. And those are results to our poll.
Very good. Okay, any other questions from the audience before we wrap up the webinar?
I don't see any.
Fantastic. Yeah. Well, with this, we're coming to the end of the webinar. Tomorrow, we will have another webinar targeted towards restarting the IVF practice. We're going to be discussing, we have a speaker and a panel that we're going to be, that will be discussing how clinics would start the IVF again. Stay tuned, and we will see you tomorrow at 3 p.m. Pacific time. Hope to see you there. Thank you for attending. I will see you then. Bye-bye.